right, we're going to give it just a few minutes and then we'll get started. All right. Hello, everyone, and welcome. Thank you for attending our webinar today on Neuroprotective Elovanoids, investigating very long-chain polyunsaturated fatty acids and their metabolites, presented by Lardon. I'm Amanda Coulter, Marketing Manager for Lardon and the host for today's webinar. Throughout the webinar, please feel free to type your questions in the Q&A section. It should be located at either the top or the bottom of your screen, and we will address these at the end of the session. I would like to start by welcoming and introducing our speakers for today. First, we are delighted to welcome guest keynote speaker, Dr. Nicholas Bazan. He is the founding director of the Neuroscience Center of Excellence at the School of Medicine at LSU Health New Orleans, and the inaugural founder of the Ernest C. and Yvette C. Valeri Chair for the Research in Retinal Degeneration. Dr. Bazan is appointed to the highest academic rank in the LSU system, a Boyd Professor, and he is also a foreign adjunct professor of neuroscience for the Karolinska Institute in Stockholm, Sweden. Dr. Bazan received his medical degree from the University of Tucumán in Argentina and trained at Columbia University College of Physicians and Surgeons, the Department of Physical Medicine and Rehabilitation in New York, and at the Harvard Medical School. At the age of 26, he was appointed Assistant Professor of Biochemistry at the University of Toronto and Assistant Director of the Department of Neurochemistry at the Clark Institute of Psychiatry. He then founded a research institute and established a doctorate and Master of Science graduate programs in biochemistry in Argentina. In 1981, he moved to New Orleans to start his next adventure. Dr. Bazan has devoted his life to studying fundamental cellular and molecular events reflected as a response to one major challenge to civilization, the growing incidence and the loss of cognition and of sight. He is a pioneer in the remarkable current realization of the significance of DHA for the central nervous system, organization, functions, and pathologies. He contributed to revealing molecular principles, including novel pro-homeostatic lipid signaling, which sustains brain and retina cell integrity. Dr. Bazan's research has contributed to and been instrumental in the scientific advancement of so many areas that it would be difficult to list every one of them today. The impact of his research includes over 500 peer-reviewed publications and an incredible amount of recognition and awards. The list goes on and on. We know Dr. Bazan has plenty to keep him busy even outside his research efforts with his lovely wife, 5 children, and 14 grandchildren. So we are very grateful to have him here today to share his time with us. Before we hand it over to Dr. Bazan, I would also like to introduce our first speaker for the day, Seth Huff, who is the Director of the Americas for Lardon. He received his degree in chemistry from Lee University and master's from the University of Toledo. He has over 11 years experience in the lipids and detergents industry, and his areas of expertise include synthesis and purification of compounds, R&D, quality management, EHS, regulatory affairs, technical support, and business development. I'm going to hand it over to Seth so he can kick us off with a brief introduction of Lardon. Seth?
Thank you for that introduction, Amanda. As Amanda mentioned, I am the Director for the Americans for Lardon Research Grade Lipids. A little background on Lardon and what we do. We have been making research-grade lipids since 1963, when we were spun out of a Danish margarine manufacturer. We are a chemical manufacturer with 10 employees currently, headquartered at the Karolinska Institute Science Park in Stockholm, Sweden, which is the largest center for academic and medical research in Sweden. We offer a comprehensive catalog of all classes of lipids, from simple fatty acids and methyl esters to more complex structures like phospholipids, ceramides, sphingolipids, oxylipins, all of these kinds of things. Another highlight, we do offer a lot of custom synthesis services, which I'll expand on in a couple slides here. All of our compounds are offered at very high purity, most over 99%, for use as reference standards and research reagents at a scale of usually microgram to gram quantities, but some products we do offer in larger scale, hundreds of grams or kilograms. We offer fast and friendly service to international customers all over the globe, and also have a US sales office located here in Michigan. A little bit about our team. Our managing director is Frederick Lindgren. Our operations director is Carl Johan or Kalle Aravan. The Americas director, as we've mentioned, is myself, and our customer service team is Emma, our customer service manager, Annette, Selena, and Rocha. Our manufacturing department is Mats, Dimitro, and Kate. If you'd like, you can browse our catalog online, our newly refreshed catalog, thanks to Amanda and our team, at www.lardon.com/products. And you'll come to this page where you can browse by product category, or you can search by either product name, synonym, cost number, a variety of things to find what you're looking for. I would say if you don't find what you're looking for, please always reach out to us. We do have a particular strength in custom synthesis of lipids, coming from predominantly our senior chemist, Professor Mats Hamburg, who spent over 5 decades at the Karolinska Institute as a researcher in the field of prostaglandins and oxylipins. His focus now is on chemical and enzymatic synthesis, but also has expertise in the isolation and purification of natural occurring lipids from a variety of sources. Our team, which is Mats and again, Kate and Dimitro, are adding about approximately 100 new compounds per year to our catalog from custom synthesis requests. Recently, these have included things like MCPDs and glycidyl esters, mixed glycerides, polar lipids, and one particularly that I'd like to tie in to Dr. Bazan's talk, which would be very long chain polyunsaturated fatty acids. We've worked on these for him and have succeeded in the synthesis of 32,6 up to 46,6 polyunsaturated fatty acids, which include some odd chain species and also omega-3 and omega-6 isomers. So by the structures you can see on the right, this is 32,6 omega-3, and then on the bottom, we have 32,6 omega-3 as well, labeled with D6. So these have been very exciting for us, and I know that Dr. Bazan has been using these in his research. And with that, I would like to hand it over to Dr. Nicholas Bazan to tell us a little more about what he's been doing with these compounds.
And then start video.
I think we need to go there.
Yeah.
Looks good.
Request remote control output. That's what, yeah.
Okay.
Okay, you
Are
should
we
be
there?
good to go.
Click one.
All right. Well, thank you so much, Amanda, for your very kind introduction, and Seth, also nice to see you. It's been a pleasure to work with you, and I'm very happy how responsive you and Lardon in Sweden were to our request to make special compounds that are not commercially available otherwise. So I'm delighted today to provide an overview about what we are doing. In fact, the target is how these novel lipids can be neuroprotective. Let me see. I just want to, you know, the way that the entire field have evolved is by the realization that there are bioactive molecules made from various lipids. On the left here, we see several compounds that are derived of arachidonic acid, and those compounds actually began at the Karolinska with the discovery of made there very early on, actually, and Ben Samuelson led all of this biology, and Matt Hamburg was that Seth mentioned, the chemist involved, and several others at the Karolinska that realized that arachidonic acid will lead to biologically active molecules. I'm not going to go into detail. It's on the left part of this comprehensive slide that we published a couple of years ago. And there, the point is that prostaglandins, for example, and the enzymes involved have opened up a number of new ways to understand medically relevant targets. On the left, you see where aspirin works, blocking that step after COX-2. And that was the discovery of Sir John Vane that also got the Nobel Prize along with Ben Samuelson years ago. Now, in the middle, you see EPA and docosahexaenoic acid. In fact, in 2004, in a major collaboration with Charles Serhan from Harvard, we identified in the central nervous system a derivative of DHA that we named together a neuroprotectin D1. And that opened up a series of ways to think about the significance of these bioactive lipids, because this is very potent neuroprotective. So up to here, we are talking about compounds that have 22 carbons. The big surprise came a few years later, and we reported this in 2017, discovering that longer chain fatty acids made also derivatives which are biologically active, and we named them Elovanoids. And I'd like to tell you the thinking behind this finding. In fact, my laboratory during the years have been looking for the molecular logic. How is it that neurons survive during prolonged periods of time, even in many cases when they are confronted to damage like in stroke, traumatic brain injury, or the onset of neurodegenerative diseases? Our thinking is mainly based on the following. Many people, well, patients will born with mutations that will lead them to familial forms of Alzheimer's. And so the symptoms, the clinical symptoms of Alzheimer's only are expressed at age 30, 40, 50. And so there are 2-3 decades in the life of those that carry the mutation in these familiar forms of the disease, when, in spite of having the mutation present, disease is not appearing clinically. And the same happens in inherited forms of retinal degeneration, like in retinitis pigmentosa. The mutation is from birth, but blindness begins 2-3 decades in life later. So why diseases do not manifest during latency in those inheritable forms of diseases? So this guided us in the design of experiments, because we began thinking about cell-specific responses that will be based on molecular principles that will modulate homeostasis disruption at the onset of the homeostatic processes. Generically, we call them uncompensated oxidative stress. So the concept that guided many of our experiments was that the brain and retina, part of the central nervous system, under imminent homeostasis disturbances, does not give up. It activates the production of molecular guardians of cellular integrity and function. A set of them are very long-chain fatty acids, neuroprotectin D1, and Elovanoids. So here is the structure of some of those. Here we have docosahexaenoic acid. And through a 15-lipoxygenase, we have a derivative of 22 carbons, neuroprotectin D1. When this fatty acid is elongated, will end up in phospholipids as a site of storage, but then through an enzymatic process of elongation that I will show you now, will form elovanoids, which have 32 or 34 carbons in length. So the contrast is here, neuroprotectin D1, 22 carbons, and the elovanoids much longer change lengths. Now, the key here for the elovanoids is an enzyme that is really very, very interesting, because this Elovel4 is an elongase that will produce the elongation. The number one is neuronal specific. And when mutations happen, there is impaired neural development, neuronal dysfunctions, hyperexcitability seizures, mental retardation, and many other conditions, including spinocerebellar ataxia. Also, mutations that happen along here will lead to blinding diseases. Recently, in collaboration with Professor Melino from the University of Rome, we identify in the promoter of Elovel4 regulatory sites that are decision makers in tumors, in neuroblastoma tumors. This was that just came up in Oncogene at the end of 2021. And so the promoter that control the elongation of those fatty acids is very important, neuronal specific, in the central nervous system, and even outside the physiology that we are talking about when we began talking about neuroblastoma, a tumor frequently occurring in children, is a key regulator. Now, so in summary, what I just told you is that brain neurons and photoreceptors selectively express this evolutionary conserved fatty acid elongase, Elovel4, that catalyzes the synthesis of very long-chain fatty acid. So remember, docosahexaenoic acid, 22 carbons. Here we are going beyond 28 carbons. And the key controlling point is the Elovel4 to lead us into the biosynthesis of the elovanoids. Now the elovanoids, what do they do? Let's just, from the very beginning, define this. In 2017, and our laboratory defined, number one, the stereospecific structure of these lipids, and characterized them and isolated them, and demonstrated that at least they are neuroprotective in these conditions. I said at least because I will show you some other targetings in a minute. They will protect neurons when they are challenged by oxygen glucose deprivation or by overactivation of the NMDA receptor. We show they are also in vivo in experimental ischemic stroke that elovanoids are neuroprotective when given after the stroke. And also, very interesting, and I'll show you some of this data, the target is to upregulate anti-apoptotic and pro-apoptotic proteins abundance. And in the retina itself, because of the connection with blinding eye diseases, the retinal pigment epithelial cells which sustain photoreceptor integrity and function is actually driven for survival by elovanoids. Now, this was an exciting time in a way, because these two papers from our laboratory with an outstanding group of people. I just want to mention some of them. For example, Jessica was a pre-med student, and today she's in third year of medical school at Tulane. And Sean also was a pre-med student, and Aram was doing his thesis. Now he's a resident doing neurology. And this was a superb group of people. Dr. Belayev still with us, and Bhattacharjee and Gordon and Mary Audrey. But this super team of people help us actually nail down and characterize these molecules and more importantly, perhaps in the retina, to envision how this works. Because in the retina, there are two cells, the photoreceptors and the retinal pigment epithelial cells. And there is an interrelationship metabolically between these two cells in the handling of this very long chain fatty acid to produce elovanoids when needed for protection. Now, just one example. We were very much interested then and now how at the synaptic level events that lead to survival are regulated. And here what you see is neurons in a petri dish, and this stain is actually a stain that will follow the structure of the nuclei of the cells, and you see compacted nuclei here in the control, and when you add NMDA, that you activate the glutamate, the membrane, postsynaptic membrane, and then the receptor, you produce massive entrance of calcium and cell death. Okay, so when we added very low amounts, we are talking about the nanomolar amounts of elovanoid 32 or 34. Look at these two as compared with this, there's tremendous protection. So we began thinking that neuronal dysfunctions of the NMDA receptor are involved in Alzheimer's, in Huntington, in depression, in neuropathic pain, in epilepsy, in many other conditions like stroke. And so we began envisioning ways to test these possibilities. And in fact, these papers, and I'm not going to go into detail, quantify the protection that I show here at the level of these receptors. So that is a targeting of elovanoids to protect neurons from NMDA receptor-mediated excitotoxicity due to the flooding of calcium into the postsynaptic neuron when this receptor is activated. Now, this leads us actually to look specifically, how is it that this is happening? So in the bottom here, you see retinal cells subjected to damage. In fact, we call it uncompensated oxidative stress because we add large amounts of H2O2 with TNF alpha, and as a function, you see the white and red bar is the damage of the cells, and as a function of concentration of elovanoid, you can see protection. So in this system, we decided to look for proteins that could be changed, and the big surprise here was that proteins like BCL proteins that are actually protective were upregulated under these conditions by the elovanoids. And we look at several of these proteins. So the pro-apoptotic proteins and the anti-apoptotic proteins are differentially regulated. You see, for example, these proteins which are actually pro-apoptotic, the abundance is downregulated, and the others which are actually regulating in a different way, in the opposite way, are upregulated. So there is a divergence in how the pro and anti-apoptotic proteins are regulated. The other interesting finding here was that certain proteins like sirtuin and iduna are also upregulated, and I'll tell you a little bit about this because it's critical in what we are doing. This is prohibiting also upregulated. So part of the interest, and we are following this up in detail nowadays, is that a protein called sirtuin-1 is clearly upregulated by elovanoids when cells are confronted with damage. But the interest about sirtuin-1 for us nowadays is because all the targets of sirtuin-1 are involved in longevity. You know, for example, gene silencing and gene stability through histones. P53 as well, because sirtuin-1 is the acetylase that will regulate histones, P53, NF-kappa B, PGC12, also insulin sensitivity, mitochondrial biogenesis, glucose tolerance, all of these processes are in a way linked to longevity. The other target of interest is IDUNA, which is also upregulated. IDUNA is a decision maker in the driving of toxic proteins from a cell into destruction in the proteasome. And the other one, prohibiting, is a scaffolding protein in the mitochondria that is involved in neuroprotection and is an adaptor molecule in membrane signaling, a transcriptional co-regulator, is also upregulated. So the Elovanoids are targeting the abundance of certain proteins that are very critical in many functions of the cell. I just don't want to take too much time, but let me show you some additional targets. I'll show you one set of experiments where in the retina we found an additional set of targets. What happened in the retina, in retinal degenerative diseases, in the back of the retina, there is accumulation of proteins, a beta peptide, and this is called drusen. So this is equivalent or similar to what happens in Alzheimer's disease, where the senile plaques have a beta peptide and so on. So, and this produces in age-related macular degeneration, extensive damage, irreversible damage, because the photoreceptors die. So what we have asked is whether or not the Elovanoid could block what oligomeric beta peptide does in the retina. I'll show you that we found that the target here is senescence programs that will actually threaten the survival. Something that supported this concept is, look at this healthy, beautiful interaction between the retinal pigment epithelial cell and the photoreceptors. And when we ablate the receptor that captures DHA, there is retinal degeneration. This was done in work in collaboration with Rice, when we discovered that in 2015 that adiponectin receptor 1 conserved DHA in the retina. So deleting these genes, there is retinal degeneration. And what we found is that the precursors of the Elovanoids are gone. And that led us to a series of experiments. I just quickly show you something that was very revealing, because we use a model of Alzheimer's disease, the 5FAD model, and what we found is that the pathways to make these lipid mediators are in deficit before the damage to the retina happened. So this is actually the pathways. These are the pathways leading to neuroprotectin D1 and to Elovanoids, and they are altered, preceding the degeneration, and the key enzyme Elovel4 also is changing in a dramatic way before the pathology begins in the retina. So this tells us that perhaps these guardians, as I said at the beginning, of cell survival could be decision makers, because when they downregulate the formation, the motion to damage is set to go forward. We have done extensive random forest analysis, and this was published in PNAS by a gifted graduate student, can do, and applying random forest analysis and other machine learning approaches. What we found is selective changes also in how the phospholipids of the retinal pigment epithelial cell and the retina are actually distributed. So because the DHA and the very long chain fatty acids will end up in specific phospholipids in the membranes, and from there, they will be as reservoirs released when needed. So the first set of experiments I want to show you along this line is applying oligomeric beta peptide into the retina and disrupting this beautiful structure. You see the honeycomb structure of the retinal pigment epithelial cells, and when oligomeric A beta peptide is injected, they are disrupted. Remember, oligomeric A beta peptide accumulates beneath the retina in age-related macular degeneration. So in this model, we wanted to know what happens with mechanisms that could actually be decision makers of the process. One outcome of what I just showed you is the survival of the photoreceptors. These are images in a mouse model where we injected oligomeric A beta peptide, and the images that you see there are optic coherence tomography. Clinically, we use optic coherence tomography to look at the retina of a patient. And we can also do that in the retina of a mouse. Here is the disruption. This is the normal, and here is how it's disrupted by A beta peptide. And what this shows is how the oligomeric A beta peptide effect is blocked by the Elovanoids. Here is quantified. You see in red is how the photoreceptors are damaged, and here is how they are protected by the Elovanoids. The question was then, what happens with different novel targets? We uncovered that one of them are senescence gene programming. And the senescence gene programming is an interesting process because it's an homeostatic response that prevents propagation of damaged cells. It's involved in many processes. But the point here was that P16, which is a critical marker of senescence gene programming, is actually modulated by the Elovanoids upon the damage induced by the oligomeric A beta peptide. Interesting, the work of others shows that removal of P16 senescent cells extends health span and reverses senescence-associated pathologies. And the opposite, when you overexpress this, you reduce proliferation and similar, type of processes of regeneration that happen in an aged mouse. So this senescence gene programming is critical for aging of cells and for cell function. And so we look in detail and found in the retinal pigment epithelial cell also that extracellular matrix regulator like matrix metalloproteinases selectively are modulated by the Elovanoids. However, in the retina, this doesn't happen. So this is a cell-selective process that we are confronted with here that lead to the regulation of a critical process for aging. And those experiments were done in mouse. We wanted to see that happen also in humans. And so in human cells, these are human retinal cells. When we expose them to oligomeric A beta peptide, we have the secretion of something called senescence-associated secretory phenotype that is composed of TNF-alpha, IL-16, IL-18, insulin-like growth factor, a whole bunch of cell-damaging compounds. And the key experiment was to ask whether or not in those human cells, when we add the damaging oligomeric A beta peptide, if we have the same response that I show you in the mouse, and the answer is yes, will the Elovanoids protect? Yes, they do. And the genes in the human cells are similar to those that we identify in the mouse. So in conclusion of this part, what happened here is that there is a activation of these senescence-associated processes and an additional form of inflammation that is called inflammagene. It's a chronic low-grade inflammation that occurs in advanced age and during other pathological conditions. So what we found is that the Elovanoids are blocking critical steps that sustain the intercellular environment between these two cells and the integrity of the photoreceptors. And so this is a path that we are currently following to try to understand age-related macular degeneration as well as neurodegenerations in the brain. Very quickly, we have taken advantage of moving into the human retina. So we began looking at postmortem retinas from patients with age-related macular degeneration. I will not give you details, but I will just tell you that we are able to look at the macula, the center of visual acuity, and the periphery of the retina, where specific photoreceptors are located. And we are able, with a technology called MALDI, mass spectrometry, this is work with Bill Gordon in our laboratory, which demonstrate very clear cell-specific changes of the precursors of these critical molecules. So this is MALDI, and here we have LCMS confirming the structures that we are seeing under these conditions. So we have extensive unpublished, in fact, we are writing this paper for publication, where we apply what I just told you to the human retina. In a nutshell, what we are looking is that the peroxisome is critical after the fatty acids come from the diet, from the liver, and go through the pathway into the retina. But then they are critical to protect these photoreceptors. And we have experimental evidence out of the human retina to support what I just showed you. Now, the last several minutes, I'd like to share with you also unpublished work. This is unpublished work in collaboration with Professor Marianne Schultzberg from the Karolinska, where we began looking at the model of Alzheimer's, which is actually a knock-in model that has actually 3 mutations: the Arctic, the Swedish, and the Boyd-Reuter Iberian mutation. And this knock-in model is interesting because do not show the unspecificity of others. So what we decided to do here was to look in the hippocampus of the mouse for the pathway that leads to the synthesis of the Elovanoids. And we found that there is a downregulation early on in the biosynthesis of Elovanoids. And so this opened up a tremendous set of opportunities to explore in models and in patients with Alzheimer's disease about the significance of the mechanism that I just showed you. This is a paper that was published in Communication Biology in collaboration with Dr. Schultzberg, the graduate student, our common graduate student, Emre, Seren Emre, did this fantastic work because we wanted to have a proof of principle. What happened in this model if we were to administer intranasally some of the lipid mediators? The mouse, the first few months postnatal life, will lose memory. And what I will show you here very briefly is that administering a group of lipid mediators will protect the loss of memory in this mouse. Okay, the group of lipids that we decided to use were pro-resolving lipid mediators. Okay, resolving E1, RBD1, RBD2, mycin, and these are the pro-resolving lipid mediators that actually Charlie Serhan discovered and demonstrated the effectiveness in various systems. And we added also neuroprotecting D1. What we did was to add them intranasally and to study various properties, electrophysiology and histology. I'll just show you part of what we found. What you see here are microglia cells. Okay, so here you have the hippocampus, and clearly you see the blockade of the microglia appearance under these conditions, and here is the quantification. And then if you see a series of other markers, pro-inflammatory and so on, this is the wild type. In the middle are actually the knock-in, and over here, the effect of the group of pro-resolving lipid mediators that we administer intranasally on several parameters that are linked to inflammation, to inflammagen, and to cell damage. So the intranasal delivery of these pro-resolving lipid mediators rescue memory, gamma oscillation. I don't show you here, but the paper demonstrates that in this model. This led us also to go into patients with Alzheimer's disease. And we decided to look at the cerebrospinal fluid. And actually, the patients are a very interesting group because some of them are subjective cognitive impairment. They're individuals that do not have yet Alzheimer's, and the mild cognitive impairment and Alzheimer's. I don't have time to go into detail. We just published this recently and in 2022, and there is a clinical examination, radiology, and laboratory confirmation of what happened in these patients. The bottom line is that as the disease progresses, the pro-inflammatory take over and the anti-inflammatory modulate the survival. We wanted to have subjective cognitive impairment in individuals because we are very much interested about the onset of Alzheimer's. When you look at CSF and other parameters in Alzheimer's samples, you are looking more at the consequences of a complex disease, and we are interested about the onset. The other thing that we did that is unpublished is actually intranasal administration of the very long-chain polyunsaturated fatty acid that Larodan made, and we discovered that they themselves will elicit remarkable protection. So here, these are animals with ischemic stroke, rats in this case, and here you see the quantification of what happened in this critical area of the stroke called penumbra, because in humans, penumbra is the area that we can rescue, and by rescuing the penumbra, we can actually protect the brain. The core is where massive damage happened, and the goal, the therapeutic goal, is to block the conversion of cells from the core into penumbra. Here we have MRI, and this image demonstrates clearly what we are seeing. This is the damage in saline injected animals with stroke. And here we have the 32 and the 34 fatty acid. Under this condition, the 32 seems to be more effective than the 34, but this is still significant. And this is the type of data that we have. Over here, you have different regions of the brain that we have sampled. And then we have looked also at specific markers of neurons. And what this shows is that neurons specifically are protected under these conditions. But as important as the morphology or the MRI or the neurons is that the neurological recovery is remarkably protected. The taller bar here are saline injected animals. As time goes on, up to 7 days after stroke. So the stroke model is 2 hours of stroke, and then 1 hour later, and 1 day after injecting or administering intranasally, not injecting, administering intranasally the fatty acid, you see these 2 bars where the protection, neurological protection happens. So this is very encouraging because open up experimental approaches that could be in the future explored to be applicable to the clinic. Now, another interesting aspect of all of the study are the molecular mechanisms. This is actually a paper just published also from our laboratory, and where we began teasing out the molecular mechanisms. And here we have a comparison of what happened in the retina and what happened in the brain with stroke, and how the receptors involved are. What we identify here is transcription of CRL and the Win5 Frizzle receptor, which are actually clearly targets with MPD, with neuroprotectin D1. So here we have 2 sites of neuroprotectin D1 protection, and the trafficking of the receptor into the cell, both in the retina and in the brain during stroke, demonstrate clearly at the molecular level how transcriptional regulation is actually involved in the targets that neuroprotectin D1 uses for neuroprotection. Of course, we are exploring similar mechanisms with Elovanoids. We believe that there is redundancy and they will be complementary. In fact, that's what our preliminary data that I'm not showing is actually demonstrated nowadays. Other forms of stroke, which are very bad, intracerebral hemorrhage, we are exploring it with our neurologists. Actually, these are interventional neurologists. This is work done in patients. And something that happened, this is a preliminary paper just published, where we found that neuroprotectin D1 is critically altered in intracellular hemorrhage in patients. And in fact, we have a major effort trying to get a handle of these observations during the course after the intracellular hemorrhage. And one other additional exciting finding, we have a major collaboration with Dr. Rezano from Spain and Dr. Ricardo Palacios, and we found mechanisms using human nasal cells. We found a very clear mechanism that the Elovanoid 34 regulates several targets that are critical in early stages of allergy. Sure enough, SASP and senescence programs are involved, autophagy, unfolding protein response, the matrix metalloproteinases, and IL-10 in our model will go down and Elovanoid will upregulate. And so this open up actually a very interesting additional approach of what I'm talking about into allergies. This is actually a model of nasal allergy where we use allergens to trigger the allergic inflammatory response. And then the cells that we use are actually human cells in culture. These particular cells are human nasal epithelial cells exposed to the allergens. But obviously, this opens up the possibility of exploring mechanisms that could be related, for example, to asthma and the like. The final piece of experimental evidence that is very exciting, we published in 2021 that when we use the SARS-CoV-2 virus in human cells, these are human lung cells, we can actually see all the damage produced by the virus, human alveolar cells. This is the experimental design and was published in 2021. And what was revealing was that the spike protein of the virus will actually produce this, will enter into the cells at this level, and different structures of elovanoids and of fatty acid will block the entrance of the virus. We are very much interested about this, and we have current experiments using the Omicron variants, but what we are seeing now are consequences of this, both cardiovascular and neurological, and we are very much interested about the long COVID consequences. So there are at least two key pathways that I told you today. The pathway leading to neuroprotectin D1 and the pathway leading with very long chain fatty acids to the synthesis of the elovanoids. So there is a signal bifurcation in cellular protection leading to neuroprotectin D1 or to elovanoid synthesis. And the discovery of the elovanoid bring another enzyme to the picture, which is a phospholipase A1, because you need a phospholipase A1 to release the very long chain fatty acids that in the membrane are in SN1 of the phospholipid. Now, just summary, I show you cell function mediators, inflammatory immune responses, and neuroprotection. I show you that the elovanoid protect photoreceptors against oligomeric A beta peptide, targeting senescence gene programming, autophagy, and extracellular matrix integrity. I show you very briefly human studies unpublished on age-related macular degeneration, where the DHA access to a specific concern rods plays a role. Show you that elovanoid pathway is downregulated in a knock-in model of Alzheimer's. Show you this very exciting work in collaboration also with Professors Scholzberg from the Karolinska on the intranasal delivery of the pro-resolving lipid mediators that rescue memory in a model. And also in collaboration with the Karolinska, this exciting work on the CSF of individuals with subjective cognitive impairment that open a new approach to try to understand the onset of the changes of the lipid mediators and the significance in Alzheimer's and also in ischemic stroke. And the MPD1 in patients with hemorrhagic stroke interests us a lot, and we have a major effort. Novel signaling on the transcriptional regulation involved in making all of this happen for neuroprotection, the allergy implications and the viral implication that really concern us now, very much so, the long COVIDs, brain and cardiovascular system. So additional future research will be to apply this to other pathologies. And perhaps, in general, inflammatory immune modulatory bioactivities, and perhaps consider applying several of these concepts to commercial potential, like medicinal foods, intracellular formulations, and the like. I just want to give a little answer to the initial question. Is there a molecular logic for neuronal life, long life? Yes. Which are those molecules? Very likely the molecular guardians do include this type of lipid mediators that we talk. And we are exploring novel targets. And we have novel information. How, for example, epigenomic events are involved, including telomerase and netosis and tau phosphorylation and mesortin. Just a final reflection, I believe there is resiliency in the central nervous system, and resiliency could be manifested in what we can call long, successful life. I believe music, art, lifestyle play a defined role in exceptional longevity and high quality of life. We can call that successful aging. And perhaps in the brain of those will be a high activity of those molecular guardians that I have referred to today and others to be discovered. So searching for the molecular logic and the molecular principles of senescence and aging relevant to sight and cognition led us to several unexpected findings, and we are actively enjoying this journey that we are embarked. I just want to thank several collaborators, the names I mentioned today during this talk, and especially Professor Marian Schulzberg from the Karolinska and Andy Obenhaus, that is our collaborator on MRI of some of the studies that I show you, and many organizations that support our work, particularly the NIH and the like. So thank you, and I'll be delighted to address questions.
Great. Thank you so much, Dr. Bazan. That was delightful. We do have some questions. I'm going to ask Seth to turn on his camera and unmute in case we have any for him as well. All right. And please feel free to continue to type your questions in the Q&A box as we're answering some of these. I just want to let everyone know Seth's contact information as well if you have questions about Lardon, and we will share the recording of this with everyone that was registered and attended. So, all right, let's get started here. So it says, does the addition of high amounts of DHA alone replicate the protective effect of Elovanoids? Excuse me.
Okay. Would you like me to answer that question?
Yes, please.
Well, okay. Yes, there are some black boxes that we are trying to reveal and define. The very first one is when we ingest fish oils or DHA in any form, the very first barrier is the microbiome. And we do not understand how the microbiome play a decision making. In fact, we have some new information in the absorption. Second, in 1989, we defined that after the absorption of DHA, the liver makes the decision of incorporated into phospholipids, and then that liver metabolized DHA will circulate in the bloodstream, and some of that will be taken up in the central nervous system and in other cells through receptor-mediated mechanisms. So there are several steps for the process of getting the DHA incorporated. Insofar as the connection with the DHA and the Elovanoids is a very important question that you ask, because then we have this Elovel-4. I mean, so before, in addition to all of that with DHA, we have this enzyme Elovel-4 that will decide to elongate the fatty acid to have them available in order to make Elovanoids. And there are many regulatory points that we are and others actively studying in order to understand how efficient that step and that conversion will be. I hope I answered the question.
Right. So speaking of Elovanoids, if high amounts of DHA don't protect, would upregulation of Elovanoids and DHA work?
I'm sorry, repeat the question, Amanda.
If high amounts of DHA don't protect, would upregulation of Elovanoids and DHA work?
Yeah, well, we don't have an answer to that yet, because the transcription regulation of Elovanoids is an important key piece. Also remember that it's not all over. In the central nervous system, is selectively expressed in neurons, and in the retina in neurons as well as in photoreceptors. And so the regulation of the Elovanoids is very complex. It has a very complex promoter reflected in all the pathologies resulting from mutations. So we don't understand clearly, because also how much DHA will be available. And one can do experiments in cells to deal with that, but then the big question is what happened in vivo. So this is a good question that need to have an answer, an experimental answer soon.
Okay, great.
Well, if there is any follow-up or any additional ones, Amanda, and you know, if they send you questions, I will be pleased to address them.
Absolutely. There is another one here. It says, how would you formulate for intranasal delivery of very long chain polyunsaturated fatty acids?
Well, that's a big one. And we have actually some intellectual property that we have actually patented about that. And it's an important issue. So far, we are actually using very low amounts in saline type of preparation with very low amounts of ethanol to deliver them. And that's the most basic delivery. But we believe that we could have some more sophisticated micro, well, I wouldn't say the word microcapsulation, but preparation of the fatty acids. So far, the way we do it and the way we publish this is by having very little ethanol in saline. And by the way, the amounts of the very long chain fatty acids that are active intranasally are very low. And what Larodan have done for us is to put deuterium into our very long chain fatty acids, and administering them intranasally with deuterium label. We can then, as a function of time, follow the very long chain fatty acid label with deuterium in different areas of the brain and make sure that they have reached the brain. So that what I just mentioned is the test that we have devised in order to test effective intranasal formulations and you know, the deuterium label very long chain fatty acids.
Great. All right, well, that is all that I see for now, and we are at the end of our time perfectly. So thank you again for your time today. Thank you, Seth. It was a pleasure having you, Dr. Razan. And as I mentioned to everyone, we will distribute the links for the recording within about 24 hours. And we'll make sure that if you're okay with it, Dr. Bazan, we can share your contact information for any further questions
Absolutely.
that may follow.
Yes, yes.
Yeah.
It's been a pleasure, Amanda.
Yes, thank you all very much and everyone for joining today. And we hope to see you at our next webinar. Thank you guys very much.
Thank you.